Dear audience, the cardiovascular system is designed to organize blood flow, which ensures the transport of nutrients, oxygen, biologically active substances, waste products, as well as immune and stem cells throughout the body. It was found that the minimum distance for effective diffusion from the lumen of the vessel to the cell is from 100 to 200 micrometers. Therefore, blood vessels penetrate everywhere in the body. The areas that lack blood vessels, such as epithelial and cartilage tissues, maintain their vitality due to the diffusion of substances from the blood through nearby vessels or through intermediary fluids, such as synovial fluid or aqueous humor. Their formation is also supported by the cardiovascular system. The transport function is important, but not the only function of blood vessels. They are actively involved in thermoregulation, and also provide such important specific processes as ovulation, sloughing off the layer of the endometrium during the menstrual cycle or erection. Each component of the cardiovascular system, including the heart, has a general structural plan. They have a lumen and a wall. In most cases, the wall consists of three layers: tunica intima, tunica media, and tunica externa. These tunics are most pronounced in large vessels and the heart. On the contrary, they can be traced only conditionally in the thinnest vessels, capillaries. The tunica intima is always lined with a single layer of squamous epithelium, called endothelium, which is fused with a subendothelial layer of loose connective tissue through a basement membrane. The tunica media is formed by loose or dense fibrous connective tissue and smooth muscle tissue in different proportions, with the exception of the heart, in the middle tunica of which cardiac muscle tissue predominates. The tunica externa is formed predominantly of fibrous connective tissue and is called adventitia, with the exception of the heart, where it is covered with mesothelium and is a type of serous tunica. Blood vessels are a system of interconnected tubular structures, among which are arteries, arterioles, capillaries, venules, veins, and arteriovenous anastomoses. During embryogenesis, vessels are formed from mesenchyme, and the first of them appear in the wall of the yolk sac. Transport of blood from the heart begins with the arteries. In cross-sections, the arteries almost always have a regular round shape with a waviness inner edge and a gaping lumen. Three tunics are clearly visible in the wall. The tunica intima is lined with endothelium, followed by a subendothelial layer and an internal elastic membrane. The tunica media consists of a connective tissue base, smooth muscle tissue, as well as elastic fibers and membranes. Elastic elements make the artery wall elastic, which smooths out pressure drops, i.e., pulsation. Muscle tissue maintains the tone of the wall and changes the diameter of the lumen of the vessel, thus affecting the speed of blood flow and blood pressure. Based on the ratio of smooth muscle tissue and elastic elements, arteries are distinguished: muscular, elastic, or mixed type. The tunica externa is formed of fibrous connective tissue with a network of blood vessels and accompanying nerve fibers. The structure of the artery is related to the haemodynamic conditions in it. The arteries located near the heart experience the highest blood pressure with strong pulsation. To smooth out pulsations, elastic elements are present in their walls, and since the strength of individual elastic elements is low, to prevent rupture they are arranged in several layers and predominate in the tunica media. These are elastic arteries. These include the aorta, pulmonary trunk, common carotid and iliac arteries. Fenestrate elastic membranes have a thickness of 2-3 microns. The largest number of them in the aorta is from 50 to 75. Between the elastic membranes there are spirally oriented smooth muscle cells, the contractile function of which is weak, and they specialize in the synthesis of elastin, collagen, and components of the ground substance with a high content of sulfated glycosaminoglycans. In the peripheral arteries, the pulsation is not so great, and the pressure drops. Its maintenance is ensured by a reduction of the lumen of blood vessels due to smooth muscle tissue, which in this case predominates in the tunica media. These are muscular arteries. Typical arteries of the muscular type are the arteries of limb. Their tunica media contains from 10 to 40 densely packed layers of smooth muscle cells, oriented in a spiral, which ensures regulation of the lumen of the vessel. The coronary arteries are also classified as muscular arteries, despite their location directly in the wall of the heart. In addition to the usual components, their tunica intima contains the bundles of smooth muscle cells surrounded by an elastic membrane that provide additional regulation of blood distribution in the walls of the heart. In mixed-type arteries, haemodynamic factors have intermediate values and, accordingly, the ratio of elastic membranes and smooth muscle tissue in them is approximately the same. From the arteries, blood flows into the arterioles, first terminal, then meta-arterioles, and then into capillaries and venules. These three components form the microcirculatory bloodstream, which ultimately ensures metabolic processes in tissues, thermoregulation, and delivery of immune cells. The wall of arterioles consists of endothelium, an internal elastic membrane. This is the tunica intima. Several layers of circularly oriented smooth muscle cells. This is the tunica media. And a thin layer of loose fibrous connective tissue. This is the tunica externa. Adjacent to the wall of arterioles are unmyelinated nerve fibers and bundles of collagen fibers. The processes of endothelial cells pass through holes in the internal elastic membrane and form gap junctions with smooth muscle cells. In small arterioles there is no internal elastic membrane. Due to the development of smooth muscle cells, arterioles play a leading role in regulating blood pressure and blood flow volume throughout the microcirculatory bloodstream and, accordingly, throughout the body. In addition, at the site where the capillaries separate from the arterioles, there are clusters of circularly oriented smooth muscle cells, precapillary sphincters that regulate the local volume of blood passing through a particular capillary. The density of the capillaries following the arterioles varies greatly in different organs. Thus, per 1 cubic millimeter of the myocardium, brain, liver, or kidneys there are from 2,500 to 3,000 capillaries. In skeletal muscle there are from 300 to 1,000 capillaries. In other organs there are usually much fewer. Despite the very small thickness necessary for metabolic processes, the general structure of the vessels can be traced in the capillary wall. It consists of three thinnest layers. The inner layer is represented by endothelial cells. The middle layer, by pericytes. The outer layer equals by thin collagen and elastic fibers and adventitial cells. Endothelial cells are connected by tight junctions and contain many pinocytic vesicles involved in the transport of substances between the blood and tissues. Pericytes are branched cells that lie mosaically inside the basement membrane and participate in the regulation of the lumen of the vessel. Adventitial cells are similar in properties to fibroblasts. They synthesize extracellular substance and serve as stem cells. There are three main types of capillaries, with continuous, fenestrate, and discontinuous endothelium. Capillaries with continuous endothelium, also called somatic, are the most common type. The diameter of their lumen is up to 10 microns, which allows red blood cells, which have a diameter of about 7.5 microns, to pass through them, only lining up in a chain one after another and in close contact with the wall, which greatly facilitates the diffusion of gases. Capillaries with fenestrate endothelium are present in the capillaries of the glomerulus of the kidney, endocrine glands, intestinal villi, and the endocrine part of the pancreas. Fenestrae are thinned areas of endothelial cells with a diameter of 50 to 80 nanometers. They facilitate the transport of substances through the endothelium. Capillaries with discontinuous endothelium are also called sinusoidal capillaries, or sinusoids. A similar type of capillaries is present in the liver and hematopoietic organs. The endothelial cells in them have clefts, and the basement membrane is discontinuous. Not only large protein molecules, but also blood cells can easily pass through such gaps. The diameter of sinusoidal capillaries reaches 20 to 30 microns, and they can accumulate significant volumes of blood. From the capillaries, blood sequentially enters the postcapillary venules, thoroughfare channels, and muscle venules, then into the veins. The structure of postcapillary venules differs little from capillaries. They have a diameter from 8 to 30 microns. As in the capillaries in the wall, in addition to endothelial cells, there are pericytes, collagen and elastic fibers, and adventitial cells. The number of these elements increases as the diameter of the vessel increases. Smooth muscle cells are absent. The endothelium of postcapillary venules has histamine receptors. Histamine causes a sharp increase in the permeability of the endothelium of these vessels, which leads to edema of the surrounding tissues. Also, postcapillary venules serve as a common place for leukocytes to exit the bloodstream. Thoroughfare channels have a diameter of 30 to 50 micrometers. On the outside they are covered with a layer of collagen fibers and fibroblasts, forming a thin outer connective tissue membrane. There are also no smooth muscle cells. Muscle venules have a diameter of 50 to 100 micrometers and contain one or two layers of smooth muscle cells that do not completely cover the vessel. Endothelial cells contain a large number of actin microfilaments, which play an important role in changing cell shape. There is also a thin layer of fibrous connective tissue on the outside. On the histological specimens of the microcirculatory bloodstream, arterioles are clearly distinguishable from venules due to the abundance of smooth muscle cells, which give the arterioles a characteristic striation. You can also see the chains of red blood cells that they form by squeezing through the narrow lumens of the capillaries. Taking into account the plasticity of red blood cells, this increases the area of their contact with the walls of the capillaries and increases the efficiency of gas exchange. Veins carry blood directly to the heart. Veins, as a rule, have a larger diameter than the arteries of the same name, and with the same diameter, the vein wall is noticeably thinner. On the histological specimens, their cross-sections take on irregular shapes, and the lumen does not gape. 3 tunics are clearly visible in the wall. According to the ratio of connective tissue and muscular elements, veins are divided into 2 types, muscular and non-muscular, or fibrous, type. Muscular-type veins are the most common in the body and are divided into veins with strong, weak and intermediate muscle tissue development. The tunica intima is formed by the endothelium and subendothelial layer of loose fibrous connective tissue and a small amount of smooth muscle cells. Sometimes a poorly developed internal elastic membrane is also present. The tunica intima may form valves, and smooth muscle cells are located near their fixed edge. The tunica media consists of predominantly circularly oriented smooth muscle cells, as well as collagen and, to a lesser extent, elastic fibers. The tunica externa is formed of fibrous connective tissue, which can also contain smooth muscle cells. The thickness of the tunica externa often exceeds the tunica intima and tunica media ones combined. With weak development of smooth muscle cells, the boundaries between the membranes, as well as between them and the surrounding connective tissue, are erased, and the entire complex is perceived as one whole. For example, the superior vena cava, brachiocephalic and jugular veins are muscular veins with a weak development of smooth muscle tissue. In the tunica media of such veins there is a thin layer of circularly oriented smooth muscle cells, and in the tunica externa there are powerful bundles of collagen fibers. The inferior vena cava is a vein with a strong development of muscle tissue. In addition to the circularly oriented smooth muscle cells in the tunica media, powerful bundles of longitudinally oriented smooth muscle cells lie in the tunica externa of this vein. There is also a special type of venous vessels, venous sinusoids. They are thin-walled extensions of the venous bloodstream. Filling them with venous blood is aimed at performing not metabolic, but other functions related to hematopoietic, such as in the spleen, endocrine, such as in the adrenal gland, or supporting, such as in the dura mater and cavernous bodies. Vessels that carry blood from arteries to veins, bypassing capillaries, called arteriovenous anastomoses. They are present in almost all organs. Arteriovenous anastomoses are divided into anastomoses without special obturative devices, and anastomoses with special obturative devices. Anastomoses without special obturative devices have circularly arranged smooth myocytes at the arterial end, which, when contracting, narrow the lumen of the vessel. Anastomoses with special obturative devices, in turn, are divided into anastomosis of occluding artery type and epithelioid type. Anastomosis of occluding artery type, in addition to circularly oriented smooth muscle cells, have ridges of longitudinally oriented smooth muscle cells protruding into the lumen of the vessel in the subendothelial layer. When circularly directed smooth myocytes contract, such a roller blocks the blood flow. Anastomoses of epithelioid type contain longitudinally oriented smooth muscle cells in the tunica media, which, at the venous end, are replaced by special epithelial-like cells. These cells are able to take on water and swell, thus closing the lumen of the vessel. Anastomoses of epithelioid type can be simple or complex. Simple ones include one branch of the arteriole, and complex ones consist of several branches forming a glomerulus, surrounded by a common connective tissue membrane. Unlike anastomoses of occluding artery type, in which the blood flow is blocked due to the contraction of muscle cells, anastomoses of epithelioid type, which block the blood flow due to the permeability of cell membranes to water, do not susceptible to fatigue. To ensure regulation, the anastomoses are richly innervated. Cardiac muscle in the four chambers of the heart wall contracts rhythmically, pumping the blood through the circulatory system. The walls of all four heart chambers consist of three major layers: the internal, endocardium; the middle, myocardium; and the external, epicardium. The tunica intima of the heart, the endocardium, consists of endothelium, subendothelial, myoelastic and outer connective tissue layers. The endocardium forms the atrioventricular valves and valves of the aorta and pulmonary artery. The valves of the heart develop as a duplication of the endocardium. Their surface is covered with vascular endothelium. The base of the valves is formed by dense connective tissue containing collagen and elastic fibers and lacking blood vessels. The tunica media of the heart, the myocardium, consists of cardiac muscle and fibrous connective tissue containing vessels and nerves. Its main cells are contractile cardiomyocytes united by intercalated discs into fibers that anastomose with each other. Along with contractile cardiomyocytes, the atria contain secretory cardiomyocytes that produce atrial natriuretic factor and store it before secretion in secretory granules. Receptors for it are present on the surface of contractile cardiomyocytes, myocytes of blood vessels, endocrinocytes of the zona glomerulosa of the adrenal cortex, and cells of the endocrine system of the kidneys. It stimulates contraction of the heart muscle, regulates blood pressure, water-salt balance, and urination. The conducting system of the heart is represented by the sinoatrial node, atrioventricular node, bundle of His and bundle branches. They contain conductive cardiomyocytes of type 1, 2 and 3. In the center of the sinoatrial node lie conductive cardiomyocytes of type I, pacemaker cells, abbreviated P cells, capable of rhythmically producing contractile impulses. Outside of them are conductive cardiomyocytes of type 2, or transitional cells, which conduct impulses to conductive cardiomyocytes of type 3, or Purkinje cells, or to contractile cardiomyocytes. The atrioventricular node consists of a small number of P cells, also located in the center of the node, and numerous conducting type 2 cardiomyocytes on the outside. Purkinje cells form the bundle of His and its branches. Their function is to transmit impulses from type 2 cardiomyocytes to contractile cardiomyocytes. Type 3 cardiomyocytes form bundles, Purkinje fibers, which are most often located between the endocardium and myocardium. There are pale staining cells, larger than the adjacent contractive cardiomyocytes, with sparse, peripheral myofibrils, and much glycogen. The tunica externa of the heart, the epicardium, is represented by a layer of dense fibrous connective tissue covered with mesothelium. It passes into the heart sac, the pericardium, of a similar structure. Between the epicardium and pericardium there is a slit-like cavity filled with a small amount of fluid that performs a lubricating function. In the connective tissue of the epicardium and pericardium, fat cells and branches of the coronary vessels are found. Dear students, today we have thoroughly examined the features of the histological structure of the main parts of the human cardiovascular system. In the upcoming lectures of the private histology course, we will continue studying the structure and function of other organ systems. Stay with us for our next meetings on the HistoNavigator channel.